This video is an SOP guide for the use of ExiProgen, an automated cell-free protein synthesis and purification instrument. This shows both exterior and internal components of the ExiProgen. The power button on the exterior allows you to turn on the power, and you can monitor the real-time progress of your experiment via the touchscreen. Inside, the instrument is equipped with Plate 1 for buffer cartridge 1 and Plate 2 for buffer cartridge 2. After the synthesis and purification processes, any remaining solutions are discarded into the waste tray installed between the buffer cartridges. The instrument also includes a disposable tip rack for holding disposable tips and an elution tube rack for holding the elution tubes containing the extracted protein samples. The cell-free protein synthesis and purification process using the ExiProgen system consists of 4 main steps. The first step is to prepare the reaction mixture for protein expression. The second step is the product preparation stage, where cartridges to be loaded into the instrument are pre-punched and template DNA is loaded. Following these preparation, the ExiProgen instrument is operated. Finally, after operation is complete, the protein synthesis experiment procedure concludes upon analyzing the purified protein sample. While several products can be used for protein synthesis with ExiProgen, this video will demonstrate a protein synthesis experiment using the ExiProgen EC-Maxi Protein Synthesis Kit. The kit is divided into Kit 1 and Kit 2, each containing synthesis and purification cartridges, respectively. As shown on the screen, lay out the components needed for the experiment. The reaction block and 6-hole punch are provided with the instrument when purchased. Take out the E. coli extract, master mix, and DEPC-DW according to the number of reaction samples and thaw them slowly on ice. If needed, thaw the positive control DNA tube on ice as well. Using the 6-hole punch, punch holes in cartridges 1 and 2 according to the number of samples to be used in the experiment. First, align the punch with the first well. This will create hole in wells 1, 3, 5, 7, 9, and 11. Then, reverse the 6-hole punch and punch again to create holes in the remaining wells 2, 4, 6, 8, 10, and 12. Repeat the same procedure for buffer cartridge 2. After punching, close the lids and store buffer cartridges 1 and 2. Remove the solution from the dialysis tube and wash both inside and outside using a squeeze bottle filled with distilled water. Place tubes that have been rinsed inside and out with sterile distilled water into rows A and B of the reaction block. Fill the tubes in row B with 500 microliter of distilled water. Sequentially add the E. coli extract, master mix, DEPC treated water, and template DNA that were previously thawed on ice, and prepare the protein synthesis mixture to a final volume of 450 microliter according to the table provided. Afterwards, use the pipette to transfer the prepared mixture into the dialysis tube located in row A of the reaction block. After adding the entire solution to the tube, mix it thoroughly using the pipette. Finally, cover the reaction block with a protection cover. The storage buffer composition is as follows, except for the storage buffer included in the kit. If you have prepared a separate storage buffer suitable for the target protein, you may load it in the same manner. Add 1 milliliter of storage buffer to cartridge 2 from column G1 to column J1. With these steps, product preparation for instrument operation is complete. Now, install the prepared kit into the instrument. First, open the door of the ExiProgen instrument and pull the base plate outward. Install cartridge 2 first, followed by cartridge 1. Install the waste tray between cartridges 1 and 2. When installing the reaction block, ensure that row A faces toward the inside of the instrument and row B faces the disposable tip rack. Mount the disposable tips into row B of the disposable tip rack. Make sure the disposable tips are placed in the same columns as the holes punched in the cartridges. Finally, verify that the positions of the cartridge, sample, and tip align. Push the base plate inward until it clicks, then close the door. Turn on the instrument. Press the green power button. Enter the protocol number indicated in the manual for the product. Press OK to confirm the elution volume and temperature settings. Review the checklist screen, then press the OK button. When you press Run on the screen, the instrument operation will begin and will take approximately 25 hours to complete. The run is now complete. When the protocol is finished, the work completion screen will appear. After the run is complete, take out the base plate and then remove cartridge 1 followed by cartridge 2. For future use, close the cartridge lids and store cartridge 1 at 4 degrees Celsius and cartridge 2 at -20 degrees Celsius. Next, remove the waste tray for cleaning. Remove the reaction block containing the target protein. Finally, remove and discard the used disposable tips. Also remove the disposable tip rack for cleaning. Fully push the base plate back in until it clicks and close the door. If you will not be continuing with additional experiments, press OK and the UV lamp will operate automatically. If UV disinfection is not required, press the skip button. Finally, press the main power button on the front to shut down the instrument. If needed, during protein synthesis and purification, you may recover certain samples from cartridge 2, specifically from rows L1, L2, K1, and K2, as shown in the figure. The recovered protein sample volume will be approximately 220 to 230 microliter. For bead samples, the volume may be too small to recover directly. In this case, add 300 to 400 microliter of distilled water, mix well, and then collect the sample. The recovered samples can be analyzed via SDS page to confirm whether the target protein has been synthesized. After protein synthesis and purification, clean and store other instrument accessories as follows. For the reaction block, wipe all used wells with paper towels and store them in a storage container filled with 70% ethanol solution. For the waste tray, rinse the interior with running water, spray with 20% ethanol, wipe with paper towels, and store. Similarly, spray the disposable tip rack with 20% ethanol, wipe it with paper towels, and store. This concludes the SOP video for the use of ExiProgen. For any further inquiries regarding ExiProgen instrument operation, please contact us via email.